Hello, hey Ynessa. Okay, so update me what's going on in school. Because we technically have like 2 weeks worth of school, right? I mean, yeah, you skipped lesson last week. So, yeah. Okay. Hello, hey Ynessa. Yeah. Weather's hot and I got fed up with the hair, so yep. So, okay, what's the update? Because technically it's that 2 weeks worth of stuff, I think. Okay, so do they also cover cells and organelles? That's ribosomes. Okay. Okay, so for chemistry is separation technique. Okay, that's all. Chapter. Because previously was the whole pipette burette setup, gas collection, gas drying, sublimation. Oh, okay, that's part of separation. So crystallization. Okay, crystallization. Yeah, it can be quite a pretty experiment to do if you're doing it right to grow the crystals. So yeah, okay. Okay. First things first. Is there any deadlines, homework, upcoming tests that I have to be aware? Week 8. So today's week 3. When's week 8? Today is week. This week is week 3, right? This week is week 3. Yeah. I mean, I don't know whether you count like 2nd of Jan, which is like one day as like a week or not a week, because your school started on Friday. So okay, so worst case scenario, I treat today as week 4, 5, 6, 7, 8. That means that we're looking at week 8. Most likely, then it starts on 16th Feb. What is tested on 16th Feb? Like 16th Feb week. Did they say what is covered? WA for, so in this case, is tested for both chem and bio. Okay. Okay. But from your description, your bio seems to be going at a faster. Okay, sure. Okay, double confirm. Is there any work or any practice questions or anything? Okay. Then I think the closest things, because we have not We have actually spent all our focus on chem. Today we switch to bio, specifically to test up to diffusion osmosis. Then wouldn't that be good? I mean, since it will be like fresh. Okay. I mean that's the aim. I mean, there's a. I'll be frank. There's a possibility the school didn't teach well. There's a possibility that you, I don't know, said something and then you missed it. It didn't hurt something. I don't know. It's like then I have to make sure things are going well, man. That's my responsibility. So yeah. Okay, this is Sec 3 Pure Bio. Okay, let's give this a go. Refer to your notes if you have to. I don't know whether the school I have notes. If yes, please do. This is F and E. So what is E? What is F? Just identify those two first. Yes, F is a chloroplast. On purpose, they color it green. So yeah. Good. Function of chloroplast. Convert sunlight to into chemical energy so it can do photosynthesis. What's E? What does mitochondria do? What does respiration do? Yes, correct. But what does respiration do? Respiration produce energy. Okay. Okay, the rest I think leave it up to you. See if you can cook. Oxygen is a reactant in F. Okay, what's the reaction? What's the equation for photosynthesis and respiration? Can do what equation? Yeah, what's photosynthesis? Then in this case, what's respiration? Both of these can be asked in chemistry and biology. You have to know this. You're expected to know. So yeah. Do you know about what's reactant and what's product? All this reactant. Reactants what's in front. Whereas whatever's at the back would be product. Same thing, whatever at the back is product. Then whatever's in front is reactant. Whatever's before the arrow is reactant. After the arrow is product. Okay, then use this to solve question 1. Carbon dioxide is a reactant in. Okay. Correct. Plus E is respiration, so that's reactant. Then for F, the glucose is a product instead. Okay. Okay, good. This one is GHI. Okay, they got different thing. G has a lot of ribosomes, like rough endoplasmic reticulum. H has a lot of mitochondria. Then I is the least. So I think we can compare we can compare in terms of the mitochondria and the rough endoplasmic reticulum with ribosomes. Makes a lot of enzymes. Sure, you can figure. Yeah. What does rough endoplasmic reticulum do? So the question asked is, what's the difference between a ribosome and a rough endoplasmic reticulum? So between a ribosome and a rough endoplasmic reticulum. Yes, what are the functions for each of them? So in this case, GHI, which one has rough endoplasmic reticulum? So what's the difference between rough endoplasmic reticulum and smooth endoplasmic reticulum? Good. Then what does good? So this one has ribosome. Then what's the function of ribosome? What's the function? What does it do? Synthesize protein. Means it makes protein. Just a heads up. Enzyme is a protein. There are many types of protein. Enzyme is a type of protein. So in this case, when the cell has to secrete enzyme, there requires a lot of ribosomes. Then what about H? What's so special about H? Sorry. Ribosome synthesize protein. Enzyme is a type of protein. Then what? What's so special about H? Hmm. What does it mean? So what does mitochondria do based on the previous question that we discussed? Why is respiration needed? Because it has to produce energy. So for H requires that for H has a lot of mitochondria because H has to provide a lot of energy. In this case, more correct. That's why it's the muscle. So in this case, G has a lot of rough endoplasmic reticulum. That's why it's links to the protein. So making enzymes is a type of protein. Whereas H has a lot of muscle, requires a lot of energy. That's why it's H, mitochondria. I is just because the other two have decided. So I has nothing special actually. Ileum is basically the small intestines. You yet to learn small intestines, but no, actually, you sort of learn small intestine, but you don't learn it as ileum and duodenum. You don't learn as terms like that. Because small intestines absorb food, right? Okay, so in this case, yeah, okay. This is cell specialization. Give it a go. Certain cells have in this case, when you say certain cells, we are referring to red blood cell and xylem. Most of 90% of the time, has special things about it that's different from other cells. So, do you know something you? Mm-hmm. Do you know what's unique about it? There's no nucleus in the red blood cell. That's why it's unique. Agree. Answer is B. Okay. This is application questions for either diffusion or osmosis. I need to read to find out. Okay. Different concentration, then concentration, change in volume. So we'll remove 48 hours. Okay, 0 0.8, 0 0.0, 0.5. Is it a misprint? 1.1, so this is 0 0.5. 0 0.5. Okay, change in volume. So if it's 0 0.8 to 1, change in volume. Concentration of sucrose. Change in volume of what? Of the plants or what? Equal volume of 5 different. Okay. This is about osmosis. Plasmolyze means that. Okay, better write down. So plasmolized. Okay, plasmolized means that the cell of the plant basically lose water. Plant. Let me double check this. Plant cell. picture to draw? Yes, I have a picture to draw. Okay, plasmolyze most of the time refers to plant cell that lost water due to osmosis, and the condition is cell membrane peel away from cell wall, meaning this is a cell wall. The water leak out so much that the cell membrane peels off. So this is the cell membrane. In normal situation, right, you will have something like this. Something like that. That this is the the outside layer is the cell wall. Immediately so the outside, this outside is cell wall. This inside layer that sticks next to the cell wall is the cell membrane. And of course the nucleus. Then the cytoplasm. In our situation, the water has leaked out. It causes the cell membrane to shrink into like this weird shape, causing all this gap to form. This is called a plasmolyzed. This is called plasmolyzed. Plasmolyzed cell, you can also use it called plasmolysis. So this is a plasmolysis is what happened. This is called plasmolyzed cell. Okay, so okay, try. Turgid refers to so when it comes to a plant cell, turgid means that the cell wall is slightly swollen. Because all the water has rushed in, then the cell membrane is like a balloon expanded and causes the cell wall to be swollen. So this is called turgid. The opposite of turgid is called flaccid. Flaccid means that of course then the cell becomes shriveled up because the water has leaked out. Of course, turgid will cause the plant, or let's say this is like, oh my god, my flower. So when it stand up straight, it's called turgid. Whereas when the plant goes, then the stem becomes flaccid. So this is due to the stem lose water, so it turns flaccid. So this is turgid. So the stem has extra water. Extra water. Yeah. So the water are basically so packed in it that it's like super fat. So this is like turgid. When we say plasmolyzed, it has to be specific. It has to be very specific that cell membrane is separated from cell wall. Plasmolyzed cells is also flaccid, meaning that the plant don't have enough water support, so it's like flaccid just means that there's not enough water in cell. Is it in this case this is flaccid but not yet plasmolyzed? Right? No. Lucky I wrote it down. Plasmolyzed is what? What did I just draw in blue line? So what's plasmolyzed? Yeah, what's plasmolyzed? What's the condition for plasmolyzed? Correct. The cell membrane has to peel away from the cell wall to create gaps. Okay, nice. Oh, okay, I guess haven't yet. Okay. Done? Okay. So try. In this case, right, what does the change in volume means? No shit, Sherlock. What volume increase? Of course, but what does the volume here refers to? Volume of what? Volume of the sea, volume of the sun, volume of Pluto, what volume of what? Yeah, good question, right? You think? Oh my god, reading skill down the drain, bro. In this case, they plot the thing. So, the change in volume of solution. What does that mean? Yeah, so in this case, the volume refers to what? Oh my god, you're freaking kidding me, bro. You have no idea what's coming, my dear. You have no freaking idea. Yeah, totally AI that, right? So what happened is that you have a sucrose solution placed in a similar container. So let's pretend the container is like a beaker or something. So you have a sucrose concentration. So this is the sucrose solution. So that's like a sugar. That's a sucrose solution. Then you place plant tissue. Plant tissue dump in. The volume refers to the volume of this. So if the volume of the sucrose solution actually increase, why though? How can the volume increase? Correct. The cell lose water, therefore the volume will increase. Then, correct. Because of the sucrose concentration is so high, when the concentration is very high, it has very little water. If the solution has very little water, water will start leaking out of the potato cell. So yeah, correct. So this is what you learned before, this osmosis. So water leak out, then the volume from here then may be increased to here. I don't think you do well in life, my dear, because if reading this, your brain is already lazy, it doesn't want to read, you don't want to figure it out. I don't know what you can do in life, my dear, because the last time you checked, life is difficult, man. Unless you find yourself, I don't know, someone who's willing to pay for all your lives, you'll be fine. Else, life is difficult. And it will be even worse with AI trying to mess up with all our lives, man. So it's either you find a way to solve problems so that you solve enough problem to give you a livelihood, or die, lor. Simple as that. Life is then die. Yeah, sure. Just saying. So yeah. Question 5. This is something to do with diffusion. This one quicker, so you want to have the larger surface area. So 2 times 2 times 2 is 8. 4 times 8 times this one, you want the largest surface area to volume ratio. I think so, or else how do you know? Sure, mental calculate your ass, man. You can do that. Yeah, I mean, I wouldn't trust you to build a plane or even fly a plane if I'm on it. I don't know, man. I don't think I can trust you on anything. You couldn't even read and figure it out. Huh? Yes, that's the volume, but the surface area? All the volume is all to 8, yes. 4 times 4 times 4 plus 2 plus 2, so it's 68. Yeah, answer would be C. C would be like way bigger in surface area. Okay, looks good. Did you know about surface area to volume ratio? Okay. Correct. The larger the area, then the higher the diffusion. Okay, question 6. This I mean, given that there's like partially permeable membrane, so I think this is talking about osmosis. So inside is two partial. The main difference between partially permeable membrane is to prove that there's osmosis. Diffusion don't need osmosis. Membrane is to block things so that water can pass through. That's osmosis. So in this case, inside is high concentration, outside is slow concentration. How do you know what's inside and outside? This one. Oh, that's why I'm trying to. That's why inside is sucrose. Outside is water. So, I think between C and D. Oh, okay. This is region X, so they draw a line. So this is inside. This is okay. That's also an arrow. So this is inside. This is outside. Inside has higher concentration. Outside has lower concentration. So what's your answer? Sure, because outside has lower concentration. Okay, that's a good that's a good use of your judgment. That's good. Okay, then that's where it is, because I don't think you learned this yet, right? This is on carbohydrates, proteins, and fats. What get digested and what it becomes. It's supposed to be chapter 3 called biological molecules. Okay. 应该 you only learn like 2 chapters, I think. The school only cover 2 chapters so far. So, and as I said, right, you're doing pure biology. You realize that whatever that you learn in school versus what comes out in exams, it's quite different. Because you can learn very passively, literally without thinking, because it's just telling you facts. But if you come up with a question, then you see a lot of pattern, and the pattern is like, yeah, they are not friendly. It has a lot of thinking, or else why do you think that everyone says that pure science students are smarter? There is a reason. So if you ever if I have a student, right, that basically do pure bio and pure chem, yeah, it's same as you, then when she do Chinese New Year visiting, I think she only got like a B4. So her cousin basically said that very easy, I got A2. Well, everyone yell at him and say, idiot, you're doing combined science, bro. Pure science, combined science is different. So if anyone tells you they have good grades and then ask them, is it combined science, pure science, they tell combined, trust me, the questions you're doing are way more difficult. And as a result, it also means that you have to put in effort to actually analyze. There's a reason why pure science students are deemed as smarter. So, because of critical thinking. If you, to be honest, if you don't want to think, then don't take pure science. Yeah, but that's the thing. Everyone wants to take pure science. Sorry. Confirm. Okay. Cell wall and correct. Question 2. Faster? What does that mean? It means before the school ends? Uh-huh. Okay. Throw that person into the toilet. Oh. Okay, how big is the team? How many people per team? No, how many people per team? Like in your team, how? 1 3. So it's 16. Okay, so 15 people can each punch in one time. I think he will die. Oh no, chlorophyll your head. Chlorophyll. Chloroplast lah, bro. Okay, what's the difference between chlorophyll and chloroplast? Okay, yeah, then what? Okay, then that. Oh, sure. Then what's the difference? Okay, then what's the relationship between chlorophyll and chloroplast? Chloroplast produce chlorophyll. Chloroplast is the thing that produce the chemical called chlorophyll. So yes, chlorophyll captures sunlight so that it converts it into chemical energy during photosynthesis. There's a chapter you will learn one specific chapter on. Do you know how many chapters you'll be learning? 17, man. Really? I thought there's more. Because I was doing the tag. I think that's 18, bro. I don't know, 17, 18, close lah. Though you see, I mean, depends on how you break down. This is where your school is at. You teach cells and organelles and diffusion. So you have all the. Sorry, what? It's so interesting, but you don't know what your teacher's saying. No, what active transport is just the opposite of diffusion. So what? You tell me what's diffusion first. True. Correct. But then the main difference is like imagine diffusion is from the high, then the ball roll to low, right? Then if it's active transport, it's from low, you have to roll back to the high ground. So that means that when you do this, but also means that it needs energy. You need to pump in energy to roll it to a high ground. Whereas diffusion, no need energy. Yeah, you don't need energy to devise gravity, but you need energy to defy gravity. So active transport is the only, at least for your syllabus, is the only process that requires energy. So yeah. Okay. So yeah. Okay. So try. Any what? For now, I have 6 Sec 3 students, but only 3 of them doing pure, including use of follow. Most of them actually takes physics, because not every school offers bio combination. Apparently, bio is like a atas combination. Depends because also depends whether the school got enough teacher to teach bio stuff long. Because in general, if you ask me, physics tends to offer more choices, but the one is like much later. But the problem is if you say you want choices, that means that you don't know what you want. So why does it even matter? You don't know what you want, then you want everything, sure. Unless you figure out what you want, then I don't know, you give me the vibe, I think you suit poly than JC if you really ask me. Yes. St. Andrews? You can try. I didn't hear a lot of good things about St. Andrews. Okay. Put it this way. St. Andrews, my perspective of it is, if you are a good student, then St. Andrews is very suitable for you. If you are a middle or not so good student, then maybe not so good. In other words, St. Andrews quite elitist. If you do well, then they celebrate you, they support you a lot, then you do very well. But if you are struggling, then you are like lagging behind, then maybe the school's culture not so supportive. That's what I heard. And I say it's purely biased. I only have like 3 students and one of the cousin of the student who entered it that said that. So when they say something different, clearly the one that did really well is because the one, I mean, of course, had tuition and also worked super hard and did pretty well. So the school is like very supportive. whereas that didn't do so well, then the school is like, oh, you have to, then you start to like, I don't know, gaslight you and say you have to like work extra hard, you know, you're like behind already, you know, yeah, do that kind of thing. So yep, so that's that. Okay, try question 2. Then you're guessing, bro. You don't. Okay, I make it big. Huh? Because processed protein should be the ribosomes, what? Ribosomes must be the black dot. Correct. Ribosome synthesize protein. Whatever process the protein is Golgi apparatus or Golgi body. I think we mentioned that 2 weeks ago. Yes, it basically process the protein and put it into a bubble called vesicle. That you read something about pinch off or something along that line. So number 2, ribosomes can be like floating out here. Ribosomes can also attach to the rough endoplasmic reticulum. So number 2 definitely is a synthesized protein. So by that, technically you can solve it's D. Then number 4, this one you see is like a bunch of like train tracks, but without the dots. Those are Golgi apparatus. It can be smooth endoplasmic as well, but then smooth endoplasmic usually, yeah, it can be either one of them. But this one here, smooth endoplasmic is to synthesize fats, but there's no selection of synthesized fats here. And you're right. Number 1, that one is basically a vesicle, a little bubble little small bubble that contains chemicals. So when this little bubble goes to the cell membrane, it will release the chemical, so secrete substance out of the cell. So and what you're saying is like Golgi apparatus or Golgi body looks very similar to smooth endoplasmic reticulum. That is correct. Yeah, but of course they function differently. So yeah. Okay. Oh, this one got the okay. Give you a try. So this is about reading already. You have to figure out what's the difference between the poison and the not poison. Okay, so what's with the poison here? Ask. Yeah, read the bloody thing. Oh my god, yeah, 这样多 nonsense. Read this thing, oh my god. The poison here is a respiratory poison. What does it mean? F you, man. I ask you what it is, you just repeat the word, then just call it an answer. What does it mean? let me translate to you. Respiratory poison means that once you get poisoned, the cell cannot go through respiration. So what does it mean? If the cell cannot go through respiration, what does it mean? Breathe and respiration not the same lah, bro. Breathe is where you breathe in air, breathe out air. Respiration means react with oxygen. Yeah, what does it mean? Oh my god, I've been repeating this same thing for 3 freaking questions. What does respiration means? It means it cannot produce energy. We repeated this for 3 times, for 3 separate times. Respiration produce energy. So that's what, because we repeat this for mitochondria also 2 times already before this. So this is the 3rd time. That means that if it get poisoned, it cannot produce energy because there's no more respiration. It cannot produce energy. No more energy. When not poisoned, then can have energy. Then once it's poisoned, then no more energy. So once the intestine is poisoned, it doesn't have energy anymore. Therefore, it cannot have active transport. Guys, remember, active transport is the only process that uses energy. Yeah, so but anyway, you had to figure out the respiratory poison. What does it do? So that means that you have glucose and maltose. Why is it A? Huh? No, I'm asking you to explain why. Why is the answer A? 120. Why are you talking high low? Okay, when in healthy one, glucose goes through 120. But once it's poisoned, it drops to 42. What does it mean? It means that the 42 is transported with diffusion. Whereas the 120 minus 42, which is like 140, oh my god, 120 minus 42, the 78 is due to active transport. You understand what I mean? Okay, maybe I would. Yeah, of course, anything that requires more than 3 seconds to focus, you can't figure it out, of course. You're freaking goldfish, that's what it is. Oh, trust me, that's not a compliment, man. Oh my god. Okay, so in this case, right, if it's a healthy intestine, okay, so if it's a healthy intestine, right, then okay, wait a minute. Okay, so in this case, right, when it is healthy, it goes through 120. When it is poison, only 42. Poison, so when it's poison, right, how come still have 42 can pass through? Because diffusion. So some of the glucose pass through diffusion. Then in this case 120, what does it mean? It means 42 is due to diffusion. Then whereas 120 minus 42, which is 78, whereas the 78 is due to active transport. So then they add up, this two portion add up together to become 120. That means that once things is poison, the active transport part is shut down. Only 42 then now can go through. Whereas if you look at the, oh my god, if you look at the maltose, okay, if you look at the maltose doesn't really matter much, because when it is healthy 39, when it's poison is 38. That means almost all of them go past using diffusion, because poison or not doesn't affect much. So in this case yeah, you go through, so that means the maltose is both going through diffusion, whereas the glucose, this one mainly has it's active transport plus diffusion, then this one is only diffusion. But they didn't write the plus thing. Maltose don't need active transport, because with healthy, maybe just plus one, just one doesn't really matter. It could be so, but there's no low, there's no high here. They are only asking you that for the healthy intestine, 39 units of maltose is transported. For the poison, then 38 is transported. So not much difference at all. Once you poison something, you are shutting down the active transport. Poison intestine can still have diffusion. That's what I'm trying to say. So by that logic, if you don't want to analyze the thing, once it's poison, you cannot have active transport. Technically, you can just figure that out using that way. Okay. But in this case, you read the data. When it is healthy, it's 39. When it's poison, it's 38. Maltose do not depend on active transport. Or else why the number so little? From 39 change to 38 is like plus one. Doesn't really matter, bro. Whereas this one has a big boost from 42, and this one become 120. It has like, it's as if this one is 42 plus 78. We already know 42 is due to diffusion, because poison still can happen. The 78 is due to active transport. Yeah. So this can be easy. Like I said, this could. I think what you need to learn is to number one, slow down your brain, and number two, not get bored and still be able to figure things out. Because I think your brain can either go really really fast but have out of control, or go really really slow, get bored, and then lose focus. That's your issue. And yeah, that's crazy. And that's why I say that I would, I'll be frank. If there's a war, I would never trust you with anything important. You'll get me killed, man. I'll be frank. And not because you're not smart. It's because of your attitude towards details. You don't care about details until you try to. The problem is if you try, you can, and that's the frustrating part. That means you can, but you choose not to. No, that means you can and you choose not to, because it's tiring. And that's why I don't trust you, because if you can and you choose not to, that's scary. So I wouldn't trust you with anything important until you can be trusted. So yeah, so that's that. Polysaccharides is sugar. You haven't learned this. This is enzyme. Yes. Food test. You haven't learned food test. Well, why is it? Yeah. Basically carbohydrate. There's different type of. You learn. I think you learned food test before. So if you That is, if you still remember your Sec 2 stuff, I largely think that they have evaporated away. If you do remember, that means less thing to learn. If you already forgot, too bad. You have to double the effort to relearn whatever that you should have already remembered last time. That's too bad, lor. I mean, that's the price to pay. Okay, question 1. Synthesizing protein. Extracellular. Which one is Golgi body? Huh? 4 is a mitochondria. Number one is rough endoplasmic reticulum because it has all the black dots. Number 2, I think, is Golgi body. Number 4 is mitochondria. Number 5 is vesicle. To be honest, I'm not quite sure what's number 3. I suppose between 2 and 3, 1 is Golgi body, 1 is smooth endoplasmic reticulum. So from this case, 1 and 3 look so similar. So I will make the judgment this is rough endoplasmic reticulum, this is smooth endoplasmic reticulum. Then number 2 will be Golgi body. So in this case, what would be your answer? One, two, 4. 5 is vesicle. Vesicle is the last step to transport oxygen. That's right. Sorry, it's the last step. Huh? Once you synthesize, you have to wrap it in the vesicle to send out. 5 is included in the synthesizing process. Synthesize means including make, process, pack, transport. The entire thing. Yes, 1, 2, 4, and 5. Last thing. Make the thing. Yeah, I understand what I mean. Okay. Okay, second last. This is specific for red blood cell. I said they will repeat red blood cell until it's older than grandma's story. They love to ask this. Okay, that looks good. Last question. Oh my god, I think I have a... Yeah, I think you'll be surprised. This is the... I mean, okay, no spoilers. Let's see how it goes. Do you know what it means? Okay, you say yeah, okay, sure. Why is it B? It feels like a random guess. Huh? B is a mitochondria? Okay. C takes... What does takes in means? Okay. Just to highlight to you, mitochondria produce oxygen? Yeah, very good. You're very good. You in this case, right? Answer is C. Why though? To do what? Yes, it uses the oxygen. What is respiration? Finally, yes. Respiration is what happens at the mitochondria to produce energy. It will use and eat away the oxygen, therefore it has the lowest oxygen concentration. C actively use away the oxygen to produce energy. Yes, this one is learning this. Basically, you see this eats away oxygen respiration thing for 3 chapters. They overlap. So if you remember one, then 3 chapters become, the other 2 chapters become easier to learn. So that's that. Okay, then we are done for the day. Really die other than the deadline on week 8. Nothing else, right? Right? Oi. Other than week 8 WA, no any important thing, huh? Okay, we'll see. Okay, then I guess. Okay, then I guess I'll see you in a week's time then, okay? Yeah. See you, bye-bye. Bye-bye.